喂喂，一二三，一二三，喂喂，一二三，一二三，喂喂，一二三，一二三，喂喂，一二三，一二三，喂喂，一二三，一二三，喂喂，一二三，一二三，喂喂，一二三，一二三，喂喂，一二三，一二三。测试测试测试测试测试测试一二测试 这是一二三，一二，这是演讲测试一二三，一二三。
Okay. Very nice. He has gone to a restaurant. Not enough sleep, that's why.
Sensitive.
Oh.
Oh my.
Yeah, it's a long journey.
I guess you're.
Yeah, that's. I have.
This is.
Not strong.
But it's okay. Oh, yes.
Yeah, it's okay. You can. Fine. Yeah.
Oh, I made it.
You made it.
Right.
How are you?
How are you? Welcome back.
Welcome back to the They're surprised.
Okay.
I want you. Oh, you should.
Yeah, 我在。
Yeah. With Kendall. We're still collaborating.
Oh, yeah?
He has some very interesting. Cool. Cool. Yeah. I think he's trying. He's like 2 years old. He's like 2 years older. He's 2 years senior than me, so he's on the job market. Or maybe. Or this is good. Okay. This is cool. Yeah. Can maybe there's cursor. Oh, I have cursor. Okay. Yeah, it's a little too. Yeah, it's too weak.
Okay, good morning, everyone. It is my pleasure to introduce Dr. Kevin Chen. He's a postdoc fellow at the Department of Molecular, Cellular, and Developmental Biology at Yale University. And he graduated from Princeton in 2023 with PhD in neuroscience, 2024. So he's very fresh. And actually, he's a former master student at IOP and NTU. And you can find, he worked with C.K. Chen. You can find his poster presentation in the hallway if you are interested. So, and Dr. Chen's research lies at the intersection of biophysics, neuroscience, and information theory. And he has been studying how living systems navigate and decide their way, right? And he has been very productive and published more than 10 papers, and also received multiple fellowships, like John McNeil Fellowship in Princeton and NTU Presidential Award. So, okay, today he will present his talk entitled How Living Systems Find Their Way from Behavioral Dynamics to Information Flow. Please welcome Dr. Kevin Chen.
Okay, can you hear me? Thank you so much for having me here today. I'm super excited to share my work with y'all. So as Ryota's kind introduction, I'm going to talk about how living systems move around and sense their environment. So 1/3 of this work will be from my grad school, and later on, the later 2/3 will be what I've been doing in the past year as a postdoc at Yale. So I want to make it somewhat a tutorial to begin with. It's higher level. There's a very exciting subfield in my mind called physics of behavior. So other than these very sophisticated, interesting structure in biological systems, when we look at their dynamic, their behavior, it's very interesting, very active, and quote unquote can be very purposeful when you look at them. And it's ranging across scales from micron and bacteria swimming to meters in these bird flocks. And in general, if I want to define this, the objective of this field, really we want to find principles and quantitative descriptions of complex and naturalistic behavior. And this is by no means a new field. This is like more than can't really see this, but more than half a century ago, we already started quantitatively look at bacterial motion. This seminal paper from Howard Berg's lab and this book called Random Walk in Biology. And I think it's more exciting in the past 1 or 2 decades because we really have this abundant tools to quantitatively measure and quantify and track animal behavior. These are some examples where you can, in an afternoon, click on a few frames and use machine learning tools, computer vision tools to visualize and track these animal behavior, social interaction, and in more complicated terrains. And also, as I'll be showing you later, we have these new tools to control what the animal senses and to build custom statistical models to describe behavior. And other than, in addition to these more specific tools, I think on the theory side, there are these new guiding principles or higher level theory goals for us to think about animal behavior as well. These are just to name a few. If you don't understand this jargon, it's okay for now. We're going to dive into these more specific, concrete examples later. One is such as the dimensionality of behavior. Most of the time, we think about behavior as a low-dimensional subset of things, the actions that an animal can take, and it's stereotypic and it's organized hierarchically. So these are some principles that we've been thinking about in the past 1 or 2 decades more quantitatively. So there are many different type of behavior that are interesting in biological system, but there's one more specific one that I'm super excited about and been thinking about in the past 5 or 6 years. It's sensory navigation. So chemical sensing can be viewed as one of the most ancient sensory modalities. So you basically cannot find any animal that doesn't use this sensory modality. They'll sense, accumulate, gather these chemical cue and try to locomote and navigate and seek the source of this in different scales. So these are examples from rodent centimeter to fly larvae all the way to worms and bacteria in millimeter or micron scales. And it really constrains us to understand what the system is computing. So I think it's a nice window to study biological computation. And even though humans are thought to be more visually dominating primates, if you blindfold humans, humans are actually very good at sensory navigation as well. So this is back in the days, people blindfolded undergrads from UC Berkeley and asked them to track this chocolate scent of trail across the field. And they can actually do this quite nicely, and they even compare its efficiency with dogs. Of course, it's slightly slower, but it's using a very similar strategy. So altogether, really, by observing these processes, it really forces you to think about what the agent is sensing. So it forces you to think about the sensing mechanism, right? What the goal is, why it's navigating, what's the internal computation that's happening, as well as the mechanics. How do they move? What's the locomotion pattern? What are their strategies? So all in all, just by looking at sensory navigation process, you really have to answer these series of questions. So that brings to the three stories I want to talk about today. Color-coded and RGB, red, blue, and green. And these I'll do a deep dive in the first one for my graduate work in worms, and later zoom out to find these more general principles of navigational dynamics. And if we have time, I can talk about this ongoing work of probing belief-driven navigation. It's kind of weird calling studying belief in science, but I'll define what I mean by that later. Okay, so let's zoom into the first one first. So I want to introduce the worm, C. elegans. I often view this as the hydrogen atom of neuroscience because it's the most compact system that has the most compact, well-studied brain, and we know a lot about it. So its whole genome is sequenced many years ago. We know all the connections of every single neuron in its brain, and we map out all the cells. And it's less well known, but we also quantified all the possible behavior repertoire a worm can take. So on the connectome side, people have probably seen these diagrams before. You can basically treat this as a graph representation. You can trace how neurons talk to each other in this network language. And it's the behavior repertoire is also well defined, so we can classify them into different types of reverse turns and steering modes. And as an experimentalist, it's very tractable. It's like 1 millimeter large, and the life cycle is 3 days. So it really is a classic experimental biophysics type of experiments where the iteration time is fast. You can think about an experiment and do it in 3 days, and then think about a new one and do it in another 3 days. And in this 300 neuron brain, more than 10% of them are chemical sensing. So it means that you have large nodes, right? Your brain is largely dedicated to sense your environment. It doesn't have vision. It has 6 neurons that are sensing touch, but mostly it's sensing chemical cues. So in the past, people have been using this assay basically as chemotax index assay, where you put worms in the middle, and there will be a droplet of a control, so there's solvent on one side and an odor of interest on the other side, and you just count the number of worms that went to the attractant after an amount of time. So you can compute the fractional difference of worms that move to the droplet, right? This is the chemotax index. But this is a very coarse result of what the worm is doing, because we don't really know how they went there and what they're sensing. So this is one of the first papers during PhD is to come up with a novel experimental apparatus to really monitor concurrently the environment, control it, and also measure what the worm is doing. So we built this flow chamber where there are parallel tubings coming from the lower left and going out from the other side. It's vacuum sealed with an acrylic piece on top, and there's this holder that holds the plate of the worm on agar, as well as a metal holder that holds this scalable odor sensor array, where we have over 100 of these metal oxide digital sensor that can sense odor, as well as 56 thermal hydro sensors. So you basically have a full landscape that you can take a picture at 0.5 hertz, or sorry, 2 hertz, and you control this with active flow. So you keep the system at some steady state. And now you can overlay the worm crawling in this environment with this landscape that you monitor and control. So here are some examples where the worm started at green dots, and they gradually migrate up gradient and end at the red dots. Okay, so here's a video of what you can measure from this equipment. So the worm will be coming in, and then it'll make a turn towards the wrong direction, but it'll realize and make a sharp turn to correct for it. And on top of that, you can also see that it can steer and gear towards the high conservation regions. Let's play it again. So for those who are familiar with the run and tumble story of bacteria, these kind of turns are kind of like a tumble, but worms are much larger than bacteria. Its nose tip is 70 micron, and it can wiggle. It can swing its head up to like millimeter scale. So it's much larger than this diffusion limit story. So on top of this tumble strategy, it can also steer in a more deterministic manner. It has a different modality. So on the left-hand side, in this orange schematic is the tumble like behavior, and in worm literature they call it pirouettes because they dance. They almost make a dance. It's like, I think this is a term in that dancers use. It's a pirouette where you turn around and then move around. And the other term is called weather vaning because it's like a weather vane where you gradually steer to align with the gradient. So to quantify how they're using these two strategies, I'm making two plots here. So as a function of bearing angle, which is the angle between your local motion and the gradient direction over here, you can see that whenever the bearing angle is small, then the turning probability is lower. Because when you're more aligned, you don't want to turn away. And when the bearing angle is large, then the turning probability is increased. This is the standard run and tumble story that we know about. But this is a novel strategy that worm uses. We can look at the curvature of steering. So whenever the bearing angle is positive, you can see that the curvature is more negative because you want to correct for it. And whenever the bearing angle is more negative, you want to correct for it so that your curvature is more positive. So this is the weather vaning strategy. So this is nice. We build a system, we can quantify these strategies. And we can also extract these time series. That's like a static view of what the worm is doing. From the video I've shown you, we can have like hundreds or thousands worms in this arena, and we can extract all these time series of concentration they experience, the head angle, and the speed. And we can build models to describe and generate these processes. So this is a turn and steer, or pirouette and weather vaning model, where from if you plot data, the density from data measured from experiments in gray, most of the time d theta, this heading has a large mass and near zero, right? Because most of the time it's steering with some width. But if you zoom in on the right-hand side over here, you can see this large density of d theta with tips near pi negative pi, which correspond to turning away and around the opposite direction, which is the pirouette. So this is a model that describes the mixture of two strategies. But if you look more closely to what's happening in a time series, there's some interesting patterns. You can see that it seems that whenever you see one turn, it comes in bouts. Whenever you see one turn, it will turn a couple of times and then come out, right? So it's kind of natural to hypothesize that the system is hierarchical. It's not just turn or not turn. It's turn and then a bout of turn and then another bout of not turning for a longer period. There's a hierarchical time organization happening during navigation. And that's exactly what we want to explicitly model. So we developed this state-dependent pirouette and weather vaning model called STAPA. So for those who are familiar with a hidden Markov model, this is a type of input-output hidden Markov model where the state transition are input dependent. For those who think about state space models, but the schematic, the physical intuition is that normally when we observe the experimental outcome are these kinematics where you have a period of time where steering is enriched and another period of time when turning is enriched. And on top of that, there are these PAW model, the pirouette and weather vaning mixture model, where you have different mixture of what strategy you want to use. That's the strategy level description. And on top of this, there's a state switching layer on top of this that gives the system some persistence, right? So now you can be in one state, and then during that state, you'll use a particular mixture of strategies, and then generate that density of kinematics that you observe. So the lower level are these biophysical measurements, but the higher level are these these persistent states. And we can use these inference to expectation maximization, these inference method to fit this to the behavior time series, and then find out the parameters of these states. So I'll directly jump to the conclusion of what we observed after fitting this data, is that skipping away, we find two states better describe behavior. One is a turning enriched state, where you see this high density around these pi and negative pi, where they're turning a lot. And when it's in this gray steering enriched state, they steer and have this narrow mass near zero. And you can do model comparison. Compare the data. This model with states will capture this turn interval distribution as well as the speed autocorrelation, but not when the system does not have state. And lastly, you can do numerical simulation. You can generate trajectories from this stateless or two-state model and find that navigation is more efficient when you have states. And this will be, hopefully it will be out in PNAS soon. Okay, so I mentioned about this turn strategy that happens in 1 or 2 seconds, and state, as you can see here, has probably a dwell time up to 30 seconds over a minute. But there are even longer time scales. So if you put a worm in a plate, why did it want to move towards that gradient, that order in the first place, right? This whole navigation assay takes a time course around 20 minutes to 30 minutes, up to an hour. The first figure I think I showed you in the plate assay was checking where the worms are after an hour, right? So it's the learning or the decision to go persistently towards that goal is on an even longer time scale. So we can actually do this experiment. We can train worms to associate odor with food or starvation. So when it's with food, it's appetitive. When it's naive, you don't do anything. When it's aversive, you pair it with starvation. And then you can modulate empirical here. In the dashed is what you observe experimentally. You can observe how the chemotaxis index, the fraction of worms moving towards the goal, is modulated by these type of treatments. And you can fit the model that I mentioned earlier to these learning-dependent trajectories and get different parameters and generate trajectories to recover a chemotaxis index. They can capture that as well. So this is basically just going after the first jargon that I mentioned about, which is hierarchical. When you talk about behavior, you probably want to talk about what time scale you're coarse graining out, or what time scale you care about, and then describe behavior with respect to that time scale. Otherwise, a lot of the times these are very hierarchical and complex. And which relates to the mechanism. If you're interested in underlying neural code, the neural network that are controlling these states, you really have to think about which time scale you're thinking about. So I won't go into detail here, but for those who are more interested in the neuroscience part of this, this is in the paper where we show that you can perturb a neuron, but the effect will be only observed on a specific hierarchy of the behavior. Okay, so that's 6 slides for 6 years of PhD. But so maybe I can pause for 5 seconds if there are any questions. I forgot to mention that if you have any question, you can ask in the middle. It's totally fine. Or if anything should be clarified. Mm-hmm. Uh-huh. Yeah. Yeah, that's a great question. So to just to reiterate Keita's question is a lot of times we're thinking about scales, but if you really look at such a log plot of, let's say, Turing probability of dwell time, some people are looking at these long tails where, or even a power law distribution where it's kind of unclear what scale the animal is working on. So I think those are mostly done in freely moving animals, and most of these things I'm describing here are a task. So when you have a task, there's a spatial length scale, there's a time scale. So you still probably want to, I think that we're introducing length scale and time scales here by asking how they're responding to odor or how they're responding conditioning on learning. So it's slightly different in that aspect because this is not freely running. But I think there are some people looking at long tails and worms as well. And in that aspect, they're relating it to foraging, which is things like levee flights, if people heard about this. Like if you have free running agent, what is the optimal way of searching an environment? People have been doing that in worms as well. So you don't have a chemotaxis assay, but you maybe you put down these patches, and you try to look at how worms are navigating these patchy or more naturalistic environment, look at the scales there. Yeah, sorry, that's a long answer to a short question. Yeah. Yes. Oh, there's no blackboard here, but anyway, if you plot the test log likelihood as a function of state, it saturates at 2 states. That's why you choose 2 states. Yes. So we selected this with through model selection. So we have, it's like a standard machine learning technique where we have held out worms and training worms and testing worms. So we use the testing worms to build a model, and then we can see how it generalizes. And if it overfits, it will come down. A lot of times, if you have hidden Markov model, when you have more and more states, you overfit. So, but we truncate before we overfit, and then we see that just by having 2 states is describing the system quite well. So yeah. Are you asking how this generalized to bacteria, or I see? I think Keita is the expert here. Yeah. In the worm, I think there are some pheromone sensing neurons in the tail, but here all these odor are known to bind to the nose tip, which itself is much larger than the bacteria already. As I mentioned, it's 70 micron and it can wiggle around, so it's like a much larger apparatus. Even though it's the hydrogen atom of a brain, it's much larger than a hydrogen atom of a cell. Okay, so thank you for your questions. So let's dive into the second part, finding more general principles. So earlier we had a lot of prior knowledge to think about turning and steering, right? What if we know less about the system? So we want to be more unsupervised and more general. So this is what I've learned from almost 10 years ago from when I was ILP from CK, which is dynamical system approach. So we want to first build a state space of behavior actions. So if you have time trajectories of behavior actions, how do you actually define these stereotypic behavior? So one way we can do this is to build the states using delay embedding. So this is inspired by Taken's theorem, saying that if you have a complex chaotic dynamical system, you can use delay embedding to reconstruct the tractor manifold. And in more mathy terms, this manifold is proved to will be diffeomorphic, meaning that the flow around these manifold will preserve in an equation-free way the physical property of this nonlinear dynamical system. So we want to be fully data-driven, right? So we'll get these time series of behavioral kinematics, and then we'll use the maximum entropy model of transitions, which is basically a Markov process along this in this delay embedding state. So we'll be coarse graining these delay embedding space into n of these states with the kth step time window of the delay embedding steps and marching through these tau time steps. And then you can look at the spectral property of this Markov matrix, which is Pij, and then do spectral decomposition of these transition properties and find these slow modes around these manifolds. So that's very mouthful, but I can show you very concrete examples. So what are some behavior that we know of before even look at animal behavior? We can look at simple behaviors in physics textbooks, right? One is Brownian particle in a double well. So there are many timescales in this system, right? There's probably diffusion, there's interaction with the well, but the slow mode of this system is the Kramer's problem, right? Is the barrier crossing process. So you can simulate this using this Langevin equation and simulate this and find the steady state solution. But what if we treat it as an animal. We don't know the governing equation of motion, and we apply this method. So we can actually reconstruct the delay embedding state space of this Brownian particle, and then find the slowest mode using the eigen decomposition in the delay embedded state space, and then color code the time series with that eigenvector. And here you can show that really partitions the two wells out without knowledge of this physical system. You can do a similar thing to excitable system. This is a Brownian particle in two wells, where you can look at excitable system. This is a Fitou-Nagumo model, which is similar to Van der Pol oscillator, where you have this stable fixed point, and you'll pass some threshold, and then enter some unstable invariant subset. And they call this excitable excursion, where after passing some threshold, you'll traverse the state space and come back. Again, you can simulate this with these coupled equations, but you can also hand the time series simulated from this process to this the state space approach that I mentioned about earlier, in an equation-free manner, and this subthreshold and the spiking mode will pop out again. So this seems pretty promising that if you have stereotypic motif that have separation of timescales, then this approach will allow you to pull out these behavior motifs. So we're going to apply this to behavior data set. Yeah. Yes, that's the promise of Taken's theorem, that you can even do it with partial observation. So I think the original proof was with the Lorenzo tractor, which has XYZ, right? But you can actually just observe from one axis and use delay embedding to reconstruct the state space. There are some assumptions that I know of. One is it has to be mild noise. If you're strongly noise-driven, noise is high-dimensional, right? This method might fail. The other is somewhat stationary. So if your fixed point stability keeps on changing and the whole analysis is really non-stationary, you need some other prior knowledge to model it. But otherwise, to my knowledge, this empirically works quite well. And it's also a white box method. It's very interpretable. There's no black box or machine learning tools here going on here. Okay, so what is the system that we want to apply it to? I want to introduce Drosophila. So being in biophysics is awesome because sometimes you can, when you get bored of one, you can read textbook of the other. So sometimes if you read the pure physics textbook, you're not expecting to see anything related to biology. But this is a famous textbook in StatMag from Professor Goldenfeld. But when I was reading the recap of StatMag in the second chapter, I realized that he said Ising model is the Drosophila of statistical physics. So this is kind of interesting. Most of the people who are doing biophysics have this physics envy. They want to map, which is what I just did. You want to map your system to a physical system. But this is the first time seeing a physicist mapping his system to biology. It's kind of interesting. But I don't want to keep this. I kind of want to make a different analogy. If a worm is a hydrogen atom, I feel that a fly should be more like graphene, right? So it scales up and still tractable, but it's very interesting. And lastly, I give Professor Goldenfeld credit for at least using italic for this genus name, but he forgot that a genus name has to have a capital D. But it's okay. It's all good. Okay, so what do we care about flies? Flies are known to be annoying in the summer when you have rotten fruit in the kitchen, right? So we can actually just recapitulate this process in the lab. This is done before I joined the group at Yale. They can blow in this complex, play it again. They can blow in this complex smoke signal, and somehow flies care about the smoke. And these are individual flies trapped in this rig, and they'll move towards the center line and try to go upwind, right? So you can count the hit rate, which is the number of flies that ended up towards the source. They basically can localize the source. And these are all imaged with IR, so the flies actually cannot see where they're going. So this is purely local and sensory driven. So I'm going to apply the state space method that I mentioned about earlier to this exact data set. So these are trajectories in X, Y, and real space concatenated from the video that I showed earlier. And the green dot is the source location. And now you can build a state space of these time-varying trajectories of kinematics. And it's a couple hundred dimensions empirically, but you can visualize it using UMAP to 3 dimensions because we can only see in 3 dimensions. But then we color code this transition mode in this state space. So the slowest mode in this system is transitioning between stopping and walking. Surprising or unsurprising. But the reason I call it stopping and walking is, these are often unsupervised, by the way. The label is attached later on when I look into what the modes look like. So it seems that in real space, the same real space, redness is when the flies are stopping and are traversed between stopping and walking. So blue is projected back in real space and is when they're walking around. And this is the eigenvector of that transition matrix in the delay embedding space, right? It attaches with the eigenvalue, which tells you the time scale, which is 6 seconds. So this tells you that the system traverses between stopping and walking on average in 6 seconds. And you can order these slow modes and look at a different, slightly faster mode. On the time scale of 4 or 5 seconds, it seems that the flies are traversing between caring about this cone-shaped plume and not caring about it. So red is when they're not tracking, just crossing and not caring about it. And blue is when they're tracking it. And later on, there's symmetry in this system, right? There's another mode that's the symmetry breaking mode. So on a time scale of 4 seconds, the flies will be coming down or going up towards the center line. So we call these the eigenstrategies because they are on a long time scale orthogonal, but on a short time scale, the physical picture here is that in any instance, the fly will carry, right? These are like basis functions. They will carry some weights on top of these eigenmodes to carry out this navigation task, and we're basically changing coordinate and decomposing these strategies. So using this approach, you can even sample from this delay embedded state space. You can sample from them and then recapitulate most of these summary statistics we observe in data, such as velocity autocorrelation and the marginal distribution of velocity along the wind direction that we observe in flight data. And even more importantly, as I mentioned, this delay embedding state space is a couple hundred dimensions, but you can start coarse graining and lump those states that are traversing between each other. And now we are going to plot the navigation hit rate, which is the number of flies that end up near the source as a function of reduced states. And data is in green. That's the data measured hit rate. We can see that the reduced state actually performs quite well on the order of 5 states. So it's actually low dimensional, meaning that I'm not showing you what the states look like here, but it means that the flies actually take a finite number of states and they transition between them. And just by doing so, they're able to go towards the goal. So it has to be asked, right? Is this a specific thing about flies or graphenes? Or is it generalized across different, this idea of navigation modes generalizable? The answer is yes. So we're going to generalize here just in one slide. We're showing that this method applies to many different types of 2D navigational data sets. As I always hear, CK said that one figure is 1,000 words. So I have to stop here to explain more. These are different data sets from cancer cell migrating, a fly order navigation that I showed you earlier, and this is worm chemotaxis from my graduate school work. And these are 2 different bacteria, Vibrio and E. coli chemotaxis. And you can also apply this to human eye tracking. We normally don't think about it like that, but if you track human eyes, searching for patterns in 2D space is also a navigation problem, right? Because you basically have local controls. And the data structure, if you think about it, are the same. You have this time series of kinematics, and you know nothing about the governing equation of motion, but you can build the state space and look at transitions in that state space and pull out the stereotypic slow modes and color code it back in real space. So that's exactly what I'm trying to do here. So here all the gray bars, can't really see the pointer, but here all the gray bars are the physical length scale. So it ranges from micron, millimeter to degrees because this degree of your eye motion. But on top of that, you also have this mode, which is the eigenvalue of this transition matrix that I mentioned earlier, that tells you the time scale. So here you can also see the time scale showing that these cancer cells are transitioning between moving and pausing on the order of 30 seconds or an hour. And flies are slightly faster, 6 seconds. Worms are 2 seconds. and bacteria is 1 or 2 seconds, and human eye traverse between these two modes in 20 milliseconds. And lastly, I want to point out that the first mode, it seems that the first mode always pull out two main types of navigational strategy. One that's more dispersed and one that's more local. Meaning, I can unpack, meaning that one allows you to traverse across space in red, then the blue ones are, although they're different, some are called pure red, some are called pause and stop, but they're more local, meaning that they allow the agent to gather more information locally. Yeah, so these are some universal property that we've been seeing across different data sets. There are more to be done. This is a project in prep. Yes, these are all the slowest modes. There are some other modes, but these are the slowest modes that I'm plotting for you. Yes, this is from that. Yes. 2 or 3, yeah, depends on how much. So it depends on what are available. If it's a point, like if the navigational data is a centroid time series, then you basically only have velocity and angular velocity. But if you have, I think in one of these you have joints. Like in the worm, we can fit these center lines to the whole body posture. Then you really have a lot of information of what the agent is doing. Yeah. I don't have a principal proof, but my intuition is that it has to be related. Yeah, because the state space is defined, right? You cluster a state space, and the distance is some similarity metric. And if you have more information saying that, although then you have more information, then you can better tell the difference between different strategies. Yeah. Yeah. But the fact is, for most of these, at least for bacteria, I really just have the centroid. I don't really have other things. I only have d theta and speed. Well, I only have angular speed and translational speed. So that's what I'm using here. Yeah. Oh, for cancer cells, I have aspect ratio. I download this from some other cancer paper. They have aspect ratio and the cell area, etc. You have more information there, but I haven't. Yeah, that's a good point. I'll note that. I haven't looked into how much these factors matter. I assume it does because I'm not an expert here, but I assume when they pause, the size of the cell should change. Okay. Thanks. Okay. Okay. Yeah. So these are two different stories, right? The first one is deep dive and just humble worm, and the second one is trying to generalize. We want to talk about behavior modes across species and spatial temporal scales. But I want to come back to a more computational aspect of this process, so the biophysical computation during navigation. So what do I mean by that? So I think the null model of navigation really is the sensory motor transformation description. Because when we learn about bacterial chemotaxis, we always talk about this run and tumble story, which is, oh, whenever the concentration is going down, then you tumble. And when it doesn't, then you don't tumble. It really is if you think about this language as a Markovian language, which is what is the sensory input, then you take a action. That's basically this picture. And on top of that, we can talk about behavior modes, which is, I didn't tell you anything about what the agent was sensing earlier, right? That just how, if you look at these time series of actions, you can extract these behavior modes. What I really want to go after, at least in the past year, I've been thinking about are these internal states. A lot of times, these are, you can call it a memory of a system, but you can also call it internal states. It's internal because you don't really observe it. Action and sensing cues are something an experimentalist can control and observe, right? But the biological system can have some internal state that you don't observe directly until later. So that's M here. And M can have its own dynamics, right? So now this transition is dependent on M. And M can have its own dynamics. So that's what I mean by having memory. So this might be a little abstract. But let's look at a super interesting, fun, concrete example. So last summer I was at Santa Barbara. I was at KITP, this theory center. I was there not doing theory. I was doing some fun experiment with Boris. He's a famous theorist. We're working on epithelium biomechanics as well as navigation, as well as two other awesome experimental PIs, Vinky and Orit. So we were walking around this California, this beach on the west coast, and we found these. Okay, sorry, this is not the beach. This is the trash bin next to the classroom. But anyway, we found these invasive species called the Argentine ants that are lining up. I hope you can see it. They're lining up here and tracking trails. So we came up with this method to extract pheromones from some ants and paint it on a piece of paper. And we trained. We took a lot of videos and trained a deep neural network to track the ants. And you can get videos that look like this. Oh, it's not synchronized. But anyway, you can see this faint circle, and these are colored dots tracking the joints of this ant, and it just goes on and on. They'll track this circle for like 5 or 10 minutes nonstop until it evaporates or until it gets tired. So you can now look at this more macroscopic tracking behavior. It's not speeding up. This is the video speeding up because I want to finish on time. You can track its locomotion as well as this detailed antenna motion. So we're trying to hopefully wrap up that story about the antenna motion part. But I just want to share one shape that we drew. We drew many different types of shapes, seeing if the ant can track it. And this is one that's very interesting that pops out and is related to this internal state I was talking about earlier. So if you draw this type of shape and you have 3 gaps. There's actually one over here that's just smaller, you can't see it. And there's a medium gap in the middle and a large gap over here. Here we're plotting the speed in color. There's a question. Yeah, that's awesome. Yeah, that's an awesome question. So in this video, this is what do we call it? A typical good. This is a typical good ant that's very consistent. A lot of times ants will leave a little bit and then come back. So we actually get counterclockwise and clockwise one. And on top of that, the width is actually an important factor because if you look at the video, I think sorry, I can play this one. If you look at it, they'll actually go in and out because the signal to noise ratio, they call this edge tracking because the signal to noise ratio is always highest along the edge. So an ant or many animals, when they're tracking some scent, they really want to go in and out of this. So when you make it larger, they actually unintuitively somehow, the signal sometimes is lower because they will get lost in this overwhelming signal that scent that doesn't tell you any directionality. So they want to go to the edge. But sometimes it loses the edge and then meander in again. So yes, when it's larger, I'm not showing this slide, but we prove that when it's larger, they'll actually wiggle more and sometimes turn around. That is the right intuition. Okay, so here I'm plotting the speed. It seems that they'll be stopping when they're turning and then slowing down when they're trying to cross. But the most exciting result that we got was when you zoom in here. So I'm plotting this condition on tracks passing this orange regime and color coding those that pass from the left in red and those that pass from the right in blue. So you can see that whenever the ant is crossing from the left, it crossed a small one, and a medium one, and then a large one. So it's building up some kind of quote-unquote belief or some confidence that this is a meaningful period of time when I'm allowed to lose a signal, but I believe that there will be a signal in front of me. Whereas if you come in from the right, it's asymmetric. You want to turn around because you think that you completely lost it. So this is a very direct proof that a seemingly simple system can have memory or belief, right? Because back to 2 slides ago, if you build up that sensory motor transformation story that cannot explain what's happening here, if your action in these seemingly simple systems are always very Markovian and just respond to the immediate stimulus, then you cannot explain this phenomenon. So in the next few slides, I will not talk about ants. Ants are just very inspiring, but you cannot actually do more precise control experiments. So inspired by this, we want Mm-hmm. Yes, exactly. So it's a very great advice. We actually tried that, but we were confounded by our summer project. It's hand drawn, so sometimes I'm lefty. So sometimes I realize I introduce some bias. But when you look carefully, this qualitative description is we realize that when you branch, the ant will actually, if this is the bifurcation point, the ant will actually ping pong a little bit and then commit to one side, which is super interesting. That we can also use it to study what's it called? You can study decision making. And even more interesting, we drew this infinite shape loop. We wanted to go after the fact that whether or not there's a handedness memory. So if I turn left this time, it's more likely or more unlikely that I'll turn left next time. But those were mostly confounded still by the fact that it's not well controlled, and pheromone is a multi-sensory cue and a different concentration. So, but maybe next summer or this summer, it's 2026. Yeah. Mm-hmm. Yeah, there are many different ants. Yeah, there are many different ants. Or it can be the same ants. These are just in whatever. These are across time across ants. Whoever crossed this, then I put down the track. So it can be. It's from many ants. So when you cross here from the left, it means that most likely you cross smaller gaps already. So you're able to cross it. But if you cross here, you never experience a gap. So you were not able to commit and you want to turn back. Not you, ants. Yeah. So for those, I just want to make one last footnote is this eventually forms a type of ratchet, Brownian ratchet, right? The symmetry, in the end, we actually counted. In the end, most ants will be doing clockwise. Because whenever you do counterclockwise, you want to turn around, right? This is like a ratcheting effect. So in the end, if you count the number of ants, did I say worms? The ants circling this, they're mostly doing clockwise action because they cannot do counterclockwise. Yeah. Exactly. So we were actually going after some theory. So this belief can actually be quantified. So the belief is actually reflected in where you search. So you have uncertainty, you want to search larger. So it is true that in theory that Boris and others wrote down, whenever you lose signal, you're opening up a cone of belief and you're searching with respect to that belief. So it is true that this is part of the prediction as well. That's a great question. We didn't systematically look at that. Oh, I can give you a reference. The ant is 3 millimeters. So I think most of the ants can cross this with no problem. The ant's body length is 3 millimeters, right? But I think if you plot the gap as a function of crossing without thinking about memory, there's some critical lengths that they're able to spontaneously. Yeah, there should be some persistent length, which is kind of complicated. We're related to their speed as well. We plotted the speed distribution. They have this autocorrelated speed, so when they're faster, it's more likely to cross, right? Because now it's confounded by sensing and mechanics too, because you can pass a gap by just zooming over and not caring. Yeah. Sorry. There's. Yeah. Thanks. So that's why after this slide, I will not be able to do that in ants. It's slightly trickier, but from this slide on, in the next 3 slides, I'll show pretty much the exact same type of experiment, but now well controlled back in the lab, the real lab on the West Coast, on the East Coast with flies, and I'm gonna measure that. I think there's. Yeah, no worries. Yeah, I see what you're talking about. I see what you're talking about. Yeah. Yeah. That's a good point. Yeah. Yeah. I see. Yeah, we did. I haven't done so, but that's a good idea. Yeah. I can look back into the data set and look at basically the probability of transition will be as a function of a real barrier. Yeah. It's a good point. So if you're a critical reviewer looking at this is confounded by the fact that the blue one experienced some curvature that's slightly different from the red. That is true. But we wanted to make this a circular arena so that the ant doesn't run away. So, but this is not well controlled in that aspect. Yeah, there's confounded that blue is curving back because it also curved before. So I take that. That's a fair critique if that's what you're going after, right? Because blue has some curvature, so it's slightly unfair as comparison to red. So that's why. Have people heard about night science and day science? Night science is when you get inspiration, and day science is when you actually do stuff and write grants. So this is very much a night science. So we're gonna turn it back to day science. Right, day science is probably less exciting, but more robust. So let's quantify belief. Before showing stuff in actual fly experiments and data, what is belief? Right, belief is something that it's not immediately clear why you can do so if you only have a sensory signal, right? For those who are in the generation watching Indiana Jones, this is a leap of faith. You just trust that it'll happen, right? Or for those who play video games, this is Assassin's Creed. You're willing to jump down because you know it'll be safe. So this can only happen when we have memory, right? If you cover this, the system doesn't have memory. Every single instance is very Markovian. Your sensory governs your action. Then this cannot be true. So it's only when this Markovianity breaks, you really have these loops or these persistent memory state that allows you to keep the relation of sensory in action in mind or whatever biophysical implementation. Then you can better navigate. And in more StatMac terms, this will happen, right? If you have a good memory, then your entropy, the uncertainty of your transitions will be reduced, right? This can only be greater or equal to. So when you have some memory that keeps track of your past sensory in action, then you have a more certain action that you can take. This allows you to have a better strategy. So how are we going to actually implement this in terms of day science? So using fly, there are many, right? We had a lot of confound of the sensing or drawing. Now we can just change all this into light. So we can use optogenetics. So we express these opsins in the antenna of a fly. So now whenever there's red light, it will be as if there's odor, right? So now we can put these flies that have this optogenetic opsin in in between this sandwich of a glass of pieces and blow wind on one side. So now you have a naturalistic wind, but the actual odor signal is virtual. But now you have precise spatial temporal control of the exact olfactory cue. And here, just to show an example, it's slightly unclear here, but there's a fly over here. And then this is the turbulent complex plume that we took in video and then played back in virtual, and showing that the fly gradually navigates this virtual environment up to the source in the upper left. And these flies are blind, so they're not using this by red vision. They're just using this virtual odor signal to go towards the source. So the fly made it, right? This fly is here. That's the source. So rather than studying the complex plume, we want to simplify this. We just want to look at ribbons. So if I, in this system, present this ribbon, and the fly is starting gray, and it goes up, and then ends in red, when the ribbon is removed, we can suddenly remove this, right? How do they search for it? This is not really gap crossing. Well, if you're interested, you can find me later. I have gap crossing assay in the flies as well. But this is not really gap crossing, but we just want to look at what the agent does when it suddenly loses signal, right? Because if you have belief, then you have some persistent behavior that allows you to regain signal later on. So here are some diverse examples of flies starting to engage and track the ribbon, and then suddenly I remove it virtually. I remove the light signal, and then it loses signal in the orange point, and it searches for it downwind. And now I can plot, I'll align these trajectories to the point when it last experienced odor, right? Its last encounter. So all these trajectories have no sensory signal here. They have some sensory signal in the past. I'm aligning them to the wind direction and ordering them from blue to yellow using its past experience. So if the fly was in odor for longer time, it's more blue. And if a fly was in odor for a shorter time, it's in yellow. So now you can see this cone shape, right? It means that if a fly was in odor for a long time, when it loses it, it's willing to dwell around that region to wait for it because it has strong confidence that this odor should be here because I've been in the odor for a long time. Whereas if you have a short experience, then you're actually not. It's still unclear if the odor will come back, right? So that's why you can see this cone shape. It seems that it's searching for it a little bit in a larger area. So here's a normative theory for why this is happening. That was just by words. There's a more formal a construction of the theory called Bayesian search theorem, where for those who know Bayes' rule, this is similar to a calculation of a posterior, but this is calculated in time. So every time you have a new encounter, you update your belief. So that's why in the Bayesian language, this is really called a belief. This is belief in time t of where the odor is in location x. And whenever you're in location x, you have some odor detection, O, and then that detection allows you to update your past belief, which is now the prior. And each step you update this. It's kind of like what people actually use in some navigation algorithm. They actually use this type of Bayesian search theorem. So now this is a schematic where if you have a flat belief and you start having some detection, your belief will shrink towards where the actual signal is. But you can imagine having an uncertain environment if your detection has some distribution, some noisiness, then your belief is more noisy as well. Right? So that's the logic. So the uncertainty or reliability of the sensory signal would affect your later on search coverage, which reflects your uncertainty. I have one last slide, and then I'll summarize. This is, I think, an ongoing but very exciting experiment. Evidence to quantify exactly. I remember there's a question about where they're going to search how long the belief really is, and this experiment will potentially show you that. So here I'm plotting 3 types of perturbation. One is just a static ribbon I mentioned earlier. One is a spatial jitter ribbon, so it's temporally stable, but it's spatially uncertain. And on the right side, it's temporal fluctuating ribbon, so it's spatially reliable. It's the same static ribbon, but it turns on and off in time. And now we look at flies searching for these ribbons after they navigated. And now we align to the yellow dot, and this is a relaxation process, up until the last point when they had experience of each of these 3 ribbons. What is the search pattern that they search for these ribbons afterwards? So you can't really see much by eye here, but you can quantify it. So as a function of probability of returning to the last point where you observe the ribbon, as a function of time, these are ordered for these 3 types of pattern. So you can see that when the fly is navigating a temporally fluctuating signal, it's more willing to return and dwell around where the last encounter was. It makes sense because the fly is used to an environment that turns on and off stochastically. So when the signal is actually gone, the fly cannot actually know it's completely gone. It's willing to believe that the signal is still around. That's for temporal. If you look at spatial, this is lag time versus the mean squared displacement of the coverage of this search area. You can see that for flies that experience this spatially jitter environment, when you remove this environment, they're more willing to search for a larger region because they were navigating an spatially uncertain environment, so they're willing to wiggle around more in space. So this is a very nice, at least the first, to our knowledge, proof of quantifying how these uncertainty are reflected in scales with these search pattern in flies. And this can only be realized in these type of data sciences where you have full control and repeated trials in these model organisms. But it really was inspired by the ants experiments. And there's some ongoing work for the neuroscience side. You can, of course, ask where this is in the brain. And I will wrap up 1 or 2 minutes. But we start perturbing some brain regions that are known to be the compass of the fly. So the hypothesis is that at least for a spatial one, you really have to keep track of where your heading is. And when it's perturbed, then you cannot keep track of the belief of where your signal really is. So that's like a physical or neurological implementation of this actual Bayesian computation. And you can also image these neurons with 2-photon imaging. These are some ongoing work. If you're interested, you can find me later. I can show you some preliminary results. Okay, so intermediate or the actual conclusion. So lessons from worm, I show you that I think experimental biophysics are quite nice to have. These skill sets are enable us to study quantitatively these measure and quantify these sensory navigation, and have this lesson of quantifying behavior across scales from a single turn to a state to learning across hours. And I show you these dynamical system approach or equation-free approach is very nice to use to extract stereotypic behavior or low-dimensional behavior modes. And lastly, I was talking about this belief-driven navigation, which is quite interesting and still ongoing. We want to pinpoint how the agent is doing it, what's the length scale, the memory scale, and what's the neural mechanisms. And I mentioned nothing about sensing during this behavior mode story, right? So that's actually another theory story that I think I don't have time, so I actually knew that I wouldn't have time, but this will be, I'll give a presentation on this last part that I don't have time to talk about in March if people are coming to APS at Denver. So I just want to skip 4 slides and conclude more properly. So I hope the fourth story tells you that a simple organism, the hydrogen atom of brain, already exhibits complex hierarchical navigation strategies. And the second one, I covered the stereotypic behavior modes that are low dimensional that span across species and scales for navigation. And the last one are these properties of belief that enable these active systems to better navigate the environment. In my mind, I feel that the physical behavior sits in the middle. So it allows you to, after looking at these interesting phenomena, you can go both sides, right? You can, some questions are going either way early already. You can ask, like, what is the mechanism? How did the agent do that? That's more microscopic correspondence. But you can also ask question in an algorithmic sense, right? Like, what is the purpose of what is the length scale? What is the limit of the system? And I feel that just start looking at these naturalistic behavior and quantifying it is a very nice first step. And lastly, I think there's an even higher level idea, zooming out of this bigger picture of behavior. I'm very much coming from this community appreciating quantitative behavior. And for those who are interested, I encourage you to check these papers thinking about what it means to quantify behavior. And this is actually from Bill's paper. So he claimed that, and I agree, if you look back in history, most of the times you actually quantify the macroscopic behavior first before you know anything about the microscopic things, right? You know about genetics way long ago before we know the genetic codes. And we know that animals can tell color before actually figuring out the 3 cone cells, RGB. And same for neuroscience, we learned that about active potential way before we found out these ion channels. So quantifying behavior is a very nice step. Okay, last 2 slides. This navigation in science, like almost 10 years ago, I was upstairs, as Ryota mentioned earlier. Somehow miraculously, my poster is still in the hallway. It's very nice to be back. And I credit CK for teaching me that like the system probably a lot of times doesn't really matter that much. We were doing retina in frogs, brain in rodents, and heart in rodents as well, and a different system. And having a very different aspect and playing around with these ideas and re-inspiration, gathering lots of inspiration for my later work as well. So I want to thank my 2 labs and thank you for your attention.
Okay.
Yes. Yes. Yeah. I see. Direction, not location. Direction, not location. Is that where you're going? Okay. Yeah. Okay. I should write that down. Yeah, that's a good point. I think location is stricter because it's private to one animal, right? Because this is a long ribbon, and it's a high throughput experiment. So most of the time there are many flies navigating together, and when I remove it, they'll actually experience the last odor in different locations. So that's why I wanted to quantify this return probability in a private sense, that each fly should return to its own last position where it experienced. But what we're saying is also, I think should be true. We should look into the direction. So they don't share the same location, but they share the same directionality, rather than that turn back to the ribbon. Is that where you're going after? Okay. Yeah. Yeah. Yeah, but the first one is edge that's is straight, but the second one, if you look at I should plot that. The second one, if you want to navigate along it, you really need a larger crosswind displacement, right? Because if you just go straight, you'll lose it. So the fly that's willing to navigate a spatial jitter ribbon has to do this.
Okay, great. So if you want to talk to him, he will be here in the afternoon, so please come to us. Yeah, thank you very much, Dr. Chen, and once again, wonderful
Thank you.
talk.
Thank you, everyone.
Yeah, let's thank him.